Hello, curious cats, and welcome to our special episodes on the animal models that made all the discoveries possible. Because I was thinking that we should celebrate people, but also the animals that helped us make all the discoveries we did so far. So, in the first one, we had mice, and today we'll have Drosophila melanogaster, which is one of my favorite organisms. I mean, I'm biased because my PhD thesis was on them, but I will show you why they're so interesting and also quite funny. So, Drosophila melanogaster, or as we all know it, the fruit flies, may appear as common pests. They're annoying. They fly around your fruits. There's so many. It seems like there's an endless supply of them. But they have been indispensable to biological research for over a century, actually. These small insects have significantly advanced our understanding of genetics, development, and disease. This discussion explores why Drosophila is a key model organism, highlights its contributions to science, and examines the creative gene names that reflect the ingenuity of researchers. So first, let's clarify. What is a model organism, and why was a fruit fly chosen for study instead of, a larger animal that looks closer to us? So, a model organism is a non-human species used to study biological processes, such as genetics or development, because it's easy to maintain, manipulate and analyze, and also very cheap to maintain. Which, when you have very tight funds, it's a very important aspect. The fruit fly meets these criteria exceptionally well. Since the early 20th century, when Thomas Hunt Morgan began studying them at Columbia University, fruit flies have been a cornerstone of genetic research. They reproduce very quickly. They lay hundreds of eggs with a new generation every 10 to 12 days, and their small size allows researchers to house thousands in a single vial. Their genome, sequenced in 2000, contains about 14,000 genes across the chromosomes. They have 4 pairs, but one of them is the sex chromosome, and it's pretty small. So the main ones that are used and where most of the genes are the first 3 chromosomes, and they share approximately 60% of genes with humans. Yeah, you heard that right, making them relevant for studying human biology. So Morgan chose Drosophila because their visible traits, such as eye color or wing shape, revealed genetic patterns. In 1911, his team identified a white-eyed mutation linked to the X chromosome, establishing the concept of sex-linked inheritance. This discovery confirmed the Gregor Mendel's principle of heredity applied to animals, cementing Drosophila's role in genetics. Additionally, their large salivary gland chromosomes, called polytene chromosomes, are ideal for studying mutations under a microscope because the DNA is replicated multiple times, making genetic changes easier to observe. Yeah, they're so big you can actually see them under a microscope. It's amazing. Their short life cycle, ease of breeding, and low maintenance costs, often kept in simple containers with very basic food, so they eat basically yeast with some sugars. So they need protein, sugars, and nothing more. Sometimes some people keep them on banana food as well, so very easy. That makes them an efficient choice for research. The fruit fly has contributed to major breakthroughs in biology. Thomas Hunt Morgan's work on the white-eyed mutation laid the foundation for understanding chromosome-based heredity, earning him the 1933 Nobel Prize in Physiology or Medicine for his discoveries on the role of chromosomes in heredity. His lab's research clarified concepts like genetic linkage, how genes on the same chromosome are inherited together, and recombination, where DNA segments are exchanged during reproduction, forming the basis of modern genetics. In developmental biology, Drosophila revealed how genes controlled body formation. In the 1980s, Christian Nusslein-Volhard and Eric Wischaus studied fly embryos to identify genes that direct development, discovering humbugs or hawks genes that determine where body parts like wings or legs form. This work, which earned them the 1995 Nobel Prize, showed that humans also have hawks genes, highlighting shared developmental mechanisms. Drosophila also aids disease research, as about 75% of human disease-related genes have equivalents in flies, which is amazing. Scientists manipulate fly genes to model conditions like Alzheimer's, Parkinson's, or even cancer, such as by studying the RAS oncogene. In the 1970s, Seymour Benzer's team used flies to identify the period gene, which regulates circadian rhythms, contributing to a 2017 Nobel Prize for Jeffrey C. Hall, Michael Rosbash, and Michael W. Young. Flies have also been used to study addiction, with genes influencing their response to substances like alcohol. Yes, they got the flies drunk, and probably on other stuff as well. And yes, they also get addicted. So, now let's get to the very fun part that I like the most, the gene names. Because it's such an old field, researchers took the liberty to call the genes they discovered really crazy names. If you look at let's say mammalian cell biology, genes and proteins tend to have pretty boring names a lot of the times, and codes and stuff, but here they went nuts. And I will tell you about they're not just the most favorite ones I have, but I made a selection because there are literally hundreds. So they're in the Drosophila field, as I said, researchers are known for assigning creative names. They still do this even now to genes based on observable effects of mutations. So it's adding a unique and memorable aspect to genetic research, and it's very easy to remember as well. These names often reflect the phenotype or function observed when the gene is mutated, and their playful nature makes complex scientific findings more accessible. So below are some notable examples, including their backstories and significance in molecular biology. First one, cheap date. This gene increases flies' sensitivity to alcohol, causing behaviors that resemble intoxication, such as uncoordinated movement. Identified in studies of ethanol response, chip date affects the fly's nervous system and has helped researchers understand addiction pathways, which are relevant to human studies of substance abuse. Next one, Tin Man. Named after the Wizard of Oz character who lacks a heart, this gene is critical for heart development. When mutated, flies fail to develop a dorsal vessel, their equivalent of a heart. Discovered in the 1990s, Tin Man is part of the homebox family, contributing to insights into cardiac development across species, including humans. Next one, Ken and Barbie. Flies with mutations in this gene lack external genitalia, resembling the smooth anatomy of Ken and Barbie dolls. I know, very creative. Identifying during developmental studies, this gene helps researchers understand sex determination and organ formation, offering clues about reproductive biology. Next one, Shaven Baby. This gene, when mutated, results in fly embryos lacking bristles, giving them a smooth, shaven appearance. Discovered in studies of embryonic patterning, Shaven Baby regulates the formation of cuticular structures, providing insights into how genes control surface features in animals. Bruchpilot, which in German means test pilot. This gene's mutation causes flies to have defective nerve signaling, leading to poor coordination and crash landing behaviors. Yeah, they basically just cannot fly and crash face down. It's been identified in the 2000s. Bruchpilot is essential for synaptic function, aiding research into neurological disorders like epilepsy. Next one, Methuselah. Named after the biblical figure known for longevity, this gene extends the lifespan of flies when overexpressed. Discovered in aging studies, Methuselah has been used to explore genetic factors that influence longevity, with potential applications to human aging research. The next one is I'm Not Dead Yet. It's a long one. And it's inspired by a line from Monty Python and the Holy Grail. And this gene also promotes longevity when mutated, because they're not yet ready to die, I guess. It's been identified in the 1990s, and it affects metabolism, and its study has provided insights into energy regulation and lifespan extension relevant to metabolic disease. Next one, Smurf. This gene's mutation causes flies to turn blue due to defects in gut function, reminiscent of the blue cartoon characters. Found during studies of digestive physiology, Smurf helps researchers understand cellular transport and disease-related processes in the gut. Cleopatra, named for its lethal interaction with the ASP gene, mimicking the historical figure's death by snakebite, this gene affects cell survival. Discovered in genetic interaction studies, Cleopatra contributes to understanding programmed cell death or apoptosis, which is critical in cancer research. Dissatisfaction. Flies with this mutation exhibit abnormal courtship behaviors, such as reduced mating success, as if dissatisfied with their partners. Identified in behavioral genetics, this gene sheds light on neural circuits governing behavior, with implications for studying social interactions in other organisms. Hedgehog. This gene, when mutated, causes fly embryos to develop a spiky hedgehog-like appearance due to the disrupted segmentation. Discovered in the 1980s by Nusslein, Volhard, and Wischaus, hedgehog is a key signaling gene conserved in humans, and its study has advanced understanding of developmental disorders and cancers like basal cell carcinoma. And the last one I have, armadillo. Named for the segmented armor-like appearance of mutant embryos, this gene is involved in cell signaling and tissue formation. It's been identified in developmental studies. Armadillo is related to the human beta-catenin gene, which is implicated in cancer when misregulated. These gene names emerged from the Drosophila research community tradition of using descriptive and very imaginative labels to reflect the mutation's phenotype or function. Unlike the more standardized nomenclature in other fields, so as I said, for example, mouse genetics, fly researchers have embraced creativity, often drawing from literature, pop culture, or just humor. This practice began in Morgan's lab and grew as the field expanded, with names like Hedgehog and Tin Man becoming iconic in developmental biology. The playful naming helps make genetic discoveries more memorable and fosters a collaborative, approachable culture in Drosophila research, while the genes themselves have provided critical insights into development, behavior, aging, and disease. So, the fruit fly has left a profound legacy in biology. From Morgan's discoveries in heredity of Hox genes in development, disease modeling, and circadian rhythm research, fruit flies have driven scientific progress. Their sequence genome and compatibility with modern tools like CRISPR make them a versatile model for ongoing research. With 60% of their genes shared with humans, they remain a valuable tool for understanding biology. It's also pretty easy to mutate different genes and then have a line with the flies with that mutated genes that you can switch on and off. And also you can target specific cells where you can turn it on and off, which is very, very handy. The story of Drosophila reflects the power of simple organisms to answer complex questions, much like, Mario Capecchi's use of mice in gene targeting. And this is a reference to the last episode that I suggest maybe will be interesting for you to also listen to. It's about, Mario Capecchi and mice and gene targeting. So the fly, or the fruit fly, or its contribution more like, shows how persistence and creativity in science can yield transformative insights. The fruit fly has transitioned from a common insect to a cornerstone of scientific discovery. And to finish the episode, I was thinking to keep the tradition as for the main episodes about the scientists and their lives, and also add a small question for you that you can search. You can search the answer afterwards as wish, but it's basically for your curiosity. And here it is for this one. Which organism, known for its wriggling movement, first demonstrated that cells could reprogram their own genetic instructions? And a tiny hint, it's not a fly, but it shares a similar pioneering spirit in research, and I will talk about it more in the next episodes. So stay tuned if you want to know more. If not, go and do your research for yourself. If this question made you interested or more curious, and I'll see you in the next one. Stay curious. Bye.